Thank you, Elizabeth. It's my great pleasure to introduce Professor Ed Calabrese, who got his PhD at UMass Amherst back in 1973. Ed has researched extensively in host factors affecting susceptibility to pollutants. He's authored over 750 papers and more than 10 books. He is the co-editor, along with Mark Matson from NIH and Hopkins, of the recently published book Hormesis: A Revolution in Biology, Toxicology, and Medicine. Uh, Interestingly, I remember enjoying this story when I visited Ed at his office in Amherst. He credits his interest in hormesis to an experiment he did as an undergraduate back in 1966. He was he and the class were studying the peppermint plant with a growth inhibiting substance, phosphon, and the professor fully expected the phosphon to inhibit the growth. However, the plant responded by growing approximately 40% taller and leafier and than the placebo plants. And so the professor was nonplussed, not understanding this. It turned out, of course, that they had accidentally used a highly diluted form of the toxic phosphon. So that set Ed off on this adventure trying to understand the process of hormesis. Now he's has many honors. He's a member of the U.S. National Academy of Sciences. He's worked on with the NATO countries' safe drinking water committees. He's on the board of scientific counselors for the Agency for Toxic Substances and Disease Registry. He's a toxicologist by trade. He's chairman of the Biological Effects of Low-Level Exposures Committee, and he's director of the Northeast Regional Environmental Public Health Center at the University of Massachusetts. He was awarded the 2009 Marie Curie Prize for his body of work on hormesis. He's a recipient of the International Society for Cell Communication and Signaling Springer Award for 2010. He's just had an amazing fulfilling career. Recently gave a talk to 20 directors at the NIH, educating them about hormesis. And he really has directed his career over the last couple of decades to trying to understand the nature of the dose response in the low dose zone, the hormesis zone, and how it provides adaptation to organisms. He just recently published what I think will be a classic paper with Mark Matson on the catabolic anabolic cycling hormesis model of health and resilience. And I have a feeling it has great applicability to our mission at McCance in terms of brain health promotion and neurodegenerative disease prevention. So with that, we welcome you, Ed, and thank you for spending time with us.
Thank you very much. Thanks for the kind invitation, and I'm looking forward to this very much. I'd have to say that my interest now in hormesis is beyond academic. It includes it, of course, but I'm now trying to use it to keep whatever few neurons are left functioning in my brain more optimally functioning. So I'm trying to tone it to individual responsiveness here for myself. But in any case, thanks very much. Just as a slight sort of a background into this, and that is it's true that as Greg mentioned, how the idea first generated with this classroom assignment that was supposed to see inhibition back in 1966. And I stayed with that project for several years with this professor, and we ultimately did publish something on it. Never knew the term hormesis at all. It was just a low dose stimulation, high dose inhibition. Ended up doing dozens and dozens of experiments on it, and then just got in the environmental revolution that was taking place as a result of Silent Spring and other things back in the 60s. I decided I was going to not pursue the this dose response line and plans. I was going to become an insecticide toxicologist. And so I changed my, you might say my graduate school was to go in that direction, and was in the toxicology thing, still am. But in 1985, I saw a flyer that was about a conference on radiation hormesis. It was going to take place in Oakland, California in August of 85. Called up the guy and I told him, I don't know what hormesis is, but I saw a biphasic response about 20 years ago. It looks a lot like what you guys are talking about. I told him my story, and he invited me out to the conference. And I went out there, and there were a lot of people who were interested in the topic, radiation chemical wise. And as it turns out, published the proceedings in health physics, and then in 1990, I put together a group that was going to, I think I wanted them to work together to see if we could resolve whether this hormetic thing was real or not, how generalizable it was, what its applications were, and created a group entity to look forward on this. And then it took about 8 to 10 years, about 8 years really, and then a whole gush of funding came in from a variety of sources, and it allowed the development of this the thinking on it to more materialize. And I'd have to say this, that on the hormesis side, it has a history of being ignored, denied, and being associated with homeopathy, and it had a death sentence early on. It was initially created, the concept was created by this guy, Hugo Schultz, back in the 1880s. And as it turns out, when he was looking at a whole bunch of toxic agents, really to come up with looking at its effects on yeast, and in these toxic agents from formaldehyde to mercury to others. He was quite surprised, not surprised at high doses they caused toxicity, but at low concentrations they stimulated the yeast metabolism and survival. And he thought he was wrong, and he kept repeating this finding. And ultimately he said, well, I think this is right. It's reproducible. It's reproducible. He then presented in 1884 to a medical conference in Greifswald, Germany, which is the town that he was living in. And as it turns out, he made a proclamation, which he probably shouldn't have done, but he claimed that he thinks he had come up with the explanatory principle of homeopathy, and he linked his findings to homeopathy. But he was in a traditional medical institute, and so at that time in Germany there was great hostility between traditional medicine and homeopathy. Homeopathy was actually about equally powerful to traditional medicine at that time in history over there. And so what happened is that he became a persona non grata very quickly. He was, as a young academic, he was only about 32, 33 years old. He became marginalized very quickly. His idea became marginalized very quickly. And he had a lot of potential for upward mobility, but he stayed at Greifswald his entire life. He's a very smart man. His name was Hugo Schultz, and so he was fighting for respect for his idea, respect for his career, and at the same time, traditional medicine had not really settled on a dose-response relationship, but homeopathy had more or less claimed the territory of a biphasic dose response. And so when the early 1900s came on the scene, especially in England, they had some very prominent neuroscientists, and they ultimately led to the recognition of a threshold dose response relationship, and it was very much contested, and Hugo Schultz was pretty much stepped on like a cigarette butt, and his career ended somewhat unceremoniously in that sense. And there was the ascendancy of traditional medicine and the descendancy of the status of homeopathy within the biomedical community. And so what happened is that the biphasic dose response was reported by many others, but it never really consolidated very well, never was integrated very well. And and was always historically associated with homeopathy. And traditional medicine has always been at the throats of homeopathy. And so when I grew up, or many others, and you had biomedical education, you were never really shown a biphasic dose response, or if something was associated with homeopathy, it was really discredited. And so this history has been a serious history throughout really the last century. And so when I entered into this field, I have to tell you, I've been approached by, all the different homeopathic companies. I know what they do. I know how they do it. I've visited their places. I know that scene and so forth. And hormesis is a different animal. It's a biomedical, traditional sort of thing. It doesn't deal actually with extremely low doses, but it deals with low doses, and we'll talk about that as we go forth. But
Oh,
there's a history.
Ed, do you want me to share your slides?
I can do that. If I can't, then I'll ask your help. How's that?
Okay,
Give it a shot right now.
okay,
I think we should have it now.
okay.
Yeah. Okay.
Oh,
With
well,
that with that
there we
little
go. Good.
long-winded introduction, I can kind of get into this thing. But I just kind of wanted you to have a sense of a little bit of the history and where this is coming from. So I'm going to be talking about hormesis, its biomedical foundations, its medical and public health implications today. Let me see. Somehow it doesn't want to scroll down. I don't know why. Maybe it will now.
Elizabeth?
Let's see.
Thank you, Elizabeth.
I don't know why it's not allowing me to scroll.
Can you just try the arrow down on your keyboard?
Yeah, I did. That's what I was actually trying. I can try to get out and do it again, see what happens.
Okay, thank you.
Yeah. I can share this thing. Okay, we'll try that one again. And I guess that's working.
There we
Yes, it's
Okay.
working now.
Yeah, so we saw that. So now the hormesis concept is this: low doses of numerous stresses. You can name them: exercising, intermittent fasting, hypoxia, heat, cold, ionizing, non-ionizing radiation, electricity, toxins, chemicals, drugs, a whole bunch of things, anything that can stress the cell. They can stimulate a wide range of adaptive responses. These induced responses, they have the potential to profoundly affect the success of public health practices and medical interventions, things like surgeries, chemo and radiation therapies, for numerous disorders, things like heart attacks, things that are really important, like heart attack, stroke, shock, brain traumatic damage, organ transplants, and many others, actually. Stresses that trigger adaptive responses, they also offer ways to enhance healthy aging. It's very broad. To improve human performance and to prevent damage to tissues exposed later to a wide range and level of injurious stresses. And we'll come up with some examples of these. So at the center of this adaptive response, in my opinion, is this phenomenon that I'm talking about today, the phenomenon of hormesis. It's a biphasic dose response that mediates processes by which human adaptation and performance may be improved. So what is hormesis? How can it be used to achieve these public health and medical advances? And when and how should hormesis be taught to undergrads and medical students? Now this is kind of definition by Calabrese here, but and so I'm calling hormesis, it's a dose response phenomenon characterized by a low dose stimulation and a high dose inhibition. It's a non-monotonic biphasic dose response, but the key feature here is that it really has very specific dose response features. That is, it's not just any type of biphasic dose response. I might have thought that, 60 years ago, but it's not really the case. When I really started to get involved with hormesis, this is so interesting. I was developing a database for it and how to characterize it. And I thought, well, gee, this low dose stimulation, my experience with the plants back in the late 60s was that the plants only were stimulated by about 30 to 60% or so. I didn't have a broad perspective, and I was very young at that stage. And so when I was looking at this back in the 90s, 30 years almost later, I thought, well, gee, this hormetic stimulation, well, gee, maybe it could be stimulated by 500%, 600%. I wasn't sure just how it would be. And so when I began to collect hundreds and hundreds and hundreds of examples of this, I came to realize that it really had a specific dose feature that would characterize this, and this is going to play a big role in what we're going to talk about. But hormesis is set within a broad dose response framework, and there are different models to describe different events. You can call them competitive models or just different models that may be useful in different situations. One that we typically work with is the threshold model, and that's we have a dose response feature. So we have a dose going from left to right, from low to high, and this little dashed line is the control group. We'll call that 100%, however we want to do it. But we have a threshold model, and there's really very little action going on until it somehow exceeds a threshold, and then you see this response going upward. And then we have a linear model in which the response is directly proportional to the dose. And then you have the hormetic model. Well, at high doses, they seem to all be alike, but something weird happens when you reach a certain point on this dose continuum in which the response actually goes below the control group. You see a J-shaped dose response. And so the action is really the same up above, but really very different down below. And this will have all kinds of implications for which model is actually correct. Because from a regulatory point of view, EPA says, well, I think carcinogens act and mutagens act in a linear fashion, and they derive their risk assessments using a linear dose response model. If you have a non-carcinogen, non-mutagen, EPA typically assumes they act in a threshold way so that you can be exposed up to a certain level. Then something bad might happen from a tox point of view. The hormetic model is saying, well, wait a minute, you guys got it both wrong. Yeah, there's dangers at high doses, but at low doses, there's actually a reduction at risk. And so it becomes very interesting, controversial, highly debated, and the stuff of toxicological, great discussions. But in general, however, when you're taking a look at all the I'm going to say tens of thousands of examples of hormetic dose responses that I have seen, one thing that really stood out to me is that, to my great surprise, or I really didn't know, was that they have generally similar quantitative features with respect to the amplitude and the range of the stimulatory response. And you can induce hormesis directly you treat it with an agent and you follow the response over time, or it could be the result of a compensatory response, and that is following an initial disruption in homeostasis. And actually, if I go back to my story on the plant issue, there was a low dose stimulation, high dose inhibition. But when I began to study this in great detail, I took measurements every single week. And if you really followed it, the first couple of weeks, there was a dose-dependent decrease. And then after about 3 to 4 weeks, there was a reversal and there was a compensatory response. And those in the low dose zone that were actually inhibited began to grow faster, outcompeted the control group. You ended up having a biphasic dose response, as I call it, an overcompensation, a stimulation response, as it turns out. But regardless, now this is also interesting, regardless of the means of the induction, whether it's giving a chemical and you get a stimulation, giving a chemical causes toxicity, but then you get a compensatory response. This is very strange, but but the quantitative features of the dose response are similar. And that happens all the time. And that told me that basically, you are obviously having many different types of mechanisms involved, and regardless of the mechanism, you're still seeing the same quantitative features of the dose response. And this really had a profound impact on my thinking, because it led me into the belief, and I think that it's true, that there are, and we'll take a look at this, that there are constraints upon this, the magnitude of the response And I believe that it's really defining the limits of biological plasticity in different systems. Now, hormesis has been controversial in some respects because some people say, well, the low dose stimulation is beneficial. And in many cases, it is. But the low dose stimulation, in my opinion, can also be harmful depending upon the circumstance. And so I think that when you're talking about hormesis, that the issue of benefit or harm should not really be part of the definition of hormesis. It should be decoupled from that. And that the assessment should be reserved for subsequent evaluation as to the biological and ecological context of the response. And I'll try to show you an example now. For example, here we're taking a look at a chemotherapeutic agent just stylistically. And I'd have to say that, and I've seen many dozens of chemotherapeutic agents that are, anti-tumor agents designed to kill tumor cells. But if you study these in low dose areas, what you find that most of them will show a stimulation of cell proliferation. Could be tumor cell proliferation. It could be non-tumor cell proliferation, and it shows a hormetic response. It'll go up by a certain percentage and then goes down into a therapeutic zone where they would actually kill the cells that they're intended to kill. But the quantitative features of this response are really very hormetic. And you can now take a look at a drug or a whole class of drugs that increase memory in vivo systems, and you plot their dose response, and they'll also show the same quantitative features of the dose response, stimulating it in memory retention at low doses, but actually being harmful at high doses. So in the first case, we have a chemotherapeutic agent that at low concentrations is stimulating tumor cell proliferation, but killing the tumor cells at high dose. Here we have the memory drug stimulating memory at high doses, but could be harmful at higher doses. So in my opinion, they're both examples of hormesis, but you have to understand the biological situation and context with which you're dealing with. I have studied hormesis really in at least 5 different ways, through dose response patterns, statistical evaluations, replication of the findings, much work on mechanistic documentation. When I deal with large data sets, I have to get the help of biostatisticians that take me along with their simulation studies to try to evaluate the frequency of hormesis in very large data sets. And so that's a whole separate story, separate papers, but all part of this integrated evaluation. But what I've learned is that in our databases, we have many thousands of, it's really many tens of thousands of hormetic dose responses that are in there as a result of rigorous a priori entry and evaluative criteria. And just as a little backdrop here, when I first got into this, I was looking for hormetic examples, and I found many hundreds and then many thousands. And then I remembered making a presentation at a conference, and a very good biostatistician stood up and he criticized my work, and he says, you have a real flaw in the work that you've done. And I said, well, what's that? And he says all you have is you just have examples of hormesis. You don't have any entry criteria. So it's kind of like asking a baseball player, well, how many, what's your batting average? And he might say, well, I got 200 hits this season for the Red Sox. But he said, well, how many times did you get up at bat? Was it 600 or 500 or how many times up at bat? Need a batting average. And he said, you don't have that. You don't have any entry criteria. You just have evaluative criteria. And I says, oh God, you're right. And I had to go back in and I had to create a whole new data set, a whole new database in which we established very rigorous entry criteria so that we could evaluate hormesis based upon numbers of doses and other things that we were evaluating on. And as it turns out, this allowed me to come up with a batting average of hormesis, so to speak. And that is what was its frequency in the biomedical literature. And so the frequency, and we have extremely strict statistical criteria for evaluation. and so forth. And as it turns out, we came up with about approximately 40% of the dose response of the studies that we evaluated with a priori entry criteria. So that we weren't biased in what we were evaluating. They satisfied the criteria. If we had been a little bit looser with the statistical criteria, we probably could have pushed maybe 60%, that sort of thing. But I was obviously trying to get published in a good journal, and so we were very strict with the criteria, extremely strict. And so it passed okay, and was approached 40%, which was pretty good in terms of showing that this was occurring. Now, in our view, my view anyways, hormesis is a very general phenomenon, and it's independent of model. You find it in plants, microbes, invertebrates, humans, in vitro and in vivo. It really seems independent of endpoint and inducing agent. And also, curiously, doesn't really make any difference whether you're evaluating at the cell level, the organ level, and individual level. And it's always been, a very interesting challenge to try to figure out how this is all happening. Now, the key feature for me with hormesis is that the stimulation is modest. It's usually at this is at maximum. This is at maximum only 30 to 60% greater than the control group, and usually not more than 100%. And so this makes it very difficult for the to know if it's real or not, because if you have a lot of lab experience, your system could be 5, 10, 15, 20% background variability. And you're looking at something at maximum only 30 to 60%. And so when I did my first work on this was very, very instructive in some ways. My advisor, the professor, he made me actually replicate this plant study. I'm going to have to say about a dozen times. He was very skeptical. You might see this thing that goes up at maximum 30%, and there's 15% or so variability. How do you know that you how do you know that it's you can reproduce it and so forth? And many different ways I try to address this. And in that particular case, I mean, after about 12 times, I kept getting these biphasic dose responses. I was really meticulous at trying to make sure I had good randomization of plants. I tried to make sure that it was fair studies. And after 12 times, I told him I was done replicating it, that I believed it was real. And then what he told me, he said that he finally agreed with me. And but there was a lot of skepticism. And then he said, I said, well, I think we're done now. He said, well, no. He said, you just raised some new questions. He said, you've been doing this work in soil. Let's see if this chemical will also stimulate if you grow the peppermint plants in hydroponics. And so I then did the same study with the large numbers of doses in hydroponics, but avoided the soil matrix, and found the same inverted use dose response there. And that time I only had to replicate it about 6 or 7 times to satisfy him. But this idea of trying to study hormesis is not easy, because you have to really understand the biological system you're working with. You have to have a good understanding of historical control variability, and you have to make sure that you're not being deluding yourself into you think it's a real response, but it's a variable response. And also this becomes more complicated, as we'll talk about it later, fairly soon, really, tied into when you have inter-individual variability within studies and trying to detect this. Now, there's also a stimulatory range, and the stimulatory range is much more variable than the amplitude. About 75% of the studies in our database are relatively narrow, but they're within about 20-fold of the concentration or dose of the typical threshold, which I'm defining here as the NOEL, the no observed effect level, or the no observed adverse effect level. So that's like the threshold line. And then right below that threshold is where that stimulation is occurring. And it seems to occur for most studies within about a 20-fold range of dose. Now, in about 2% of our studies, the hormetic stimulation can be enormous. It can be over 1,000-fold range. And then there are some intermediaries. That in of itself has some very large implications, because if you're really trying to give somebody a hormetic dose or take a hormetic dose, you really would like to have a very wide therapeutic range so that you don't end up being too close to a toxic level. And so this has very important implications. This is a typical hormetic sort of a schematic dose response, and I'm trying to represent the dose on below on this graph increasing, and we have on the vertical where the response will be, and we have our dashed for the control. And you can see we go out a little bit down the dose line, and then we see this stimulation taking place, and I'm representing that it maxes out at around 30 to 60% above the control, and then begins to dip down, passes the control level, which I call the no observed adverse effect level, and then just shows a typical hormetic sort of a dose response. You can see this inverted U-shaped dose response. So you can see this represented with a J-shaped response, as I showed on the previous slide. It depends on the endpoint that you're looking at. If you're looking at longevity or growth, then you see this as an inverted U. If you're looking at disease incidence, it could be plotted with a J, but it's still a hormetic dose response. Now, I'm trying to represent to you a human population that's fairly diverse, so that we have a range of susceptibilities in the population. And I wrote a review paper once for the American Journal of Public Health, and I don't think this has ever happened to anybody before, but usually you're asked to write a review paper when they think you know something. And so I wrote this paper, and the guy on the editorial board then wrote a rejoinder paper in the same issue of that publication, criticizing my paper because I didn't address the issue of high-risk groups and hormesis, especially with its regulatory applications. I said, well, I guess you're right. And so it forced me to go into the database and to look to see what we could learn. And I found copious information in there when you had a wide spectrum of individual susceptibilities to the same agent. How would the dose responses display themselves? And we found that if you had the same agent, but you had normally responding subjects in a population, you get this inverted U-shaped dose response, but they would be responding at a much higher dose than those who might be more compromised, or what I call high-risk groups. But the high-risk groups would also show the hormetic dose response, but at a lower dose response, a lower dose than the normal individuals. And this has huge societal implications, because when you have such a highly diverse population as humans, you could be saying, well, gee, I'm recommending this dose because it's the optimal hormetic dose for the average person, but that dose for the average person might be toxic for somebody who is a high-risk group. And so you have to understand the nature of the population that you're working with within that framework. And so this plays into, I think, all kinds of medical treatments and things of that nature. I spent a lot of time trying to understand whether hormetic responses would be found in all different kinds of biomedical systems and pathways and activating systems and the like, and ended up publishing papers on probably all of these agents and more, and finding that hormetic dose responses occur in just about every single system that I could find biologically. I really haven't found any exceptions to this thing. And so this is just a potpourri of listing here. Many studies have provided mechanisms to account for hormetic responses. I have to tell you, back in 2000, back in 1999, I had submitted 6 papers to the Toxicological Sciences, which is the top journal in my field of toxicology. And the editor-in-chief rejected, all the papers were reviewed pretty well, but he rejected 6 papers in a row. And so I said, gosh, I said, this isn't, not a good thing. So I called up my grant manager, and I said, I have to confront this issue, right? And so I invited 7 people who had made their name on low-dose linearity, and I sent them, and I wanted to debate them. And so I sent them 150 papers on hormesis, gave them 9 months to review these things, invited them out to Amherst in January of 2000. and we had a 3-day meeting, and I gave an 8-hour presentation on hormesis. My group had 31 posters. And then they had the second day, so 7 people came up, and they all pretty much beat me up the second day. And that's where the biostatistician criticized me. But then the editor-in-chief of Tox Sciences stood up there and says, I don't care how many examples you have. He says, in toxicology, we worship at the altar of mechanism, and unless you can get a mechanism of hormesis, you're not getting in my journal. And so it was a very interesting 3-day meeting, a very helpful meeting for me. And eventually I was able to address his issues on mechanism and so forth. It took a number of years, but yeah, each mechanism is unique to the model, the tissue, the endpoint, and the agent. And there are receptor level assessments, cell signaling pathway assessments. And today, the last 15 years, hormesis has been just driven by the same quality of evaluation I see that goes on in traditional pharmacology and toxicology. So the mechanisms are at that level and are extremely complex, well studied, and also very confusing, because when you're really trying to come up with something, there's always the next paper, and the next paper forces you. You think you got the mechanism down exactly. The next paper comes in and it's a slightly, it's 2 systems are talking to each other, or 3. There's always more complexity, but the good thing about it is that there's a lot of mechanism work on hormesis, and we can go and really dig down into that at some point as well. And there are, I'd say, more than several hundred, probably 500 or so hormetic dose responses that have good mechanisms at the level of receptor signaling pathway. Really extremely good work along those lines. But regardless of the mechanism, whether it's receptor signaling, whether it's non-receptor mediated, whether it's direct or compensatory stimulation, regardless of regardless of the system that you're working with, model, the quantitative features of the dose response are the same. That's really an extraordinary thing, to think that your patterns of dose response just are not affected by your mechanism, and they're not affected by the endpoint and the agent that's inducing it, or even whether it's in a plant system or a microbial system or any other system. And you say, well, how is this even possible? And it really suggests that this hormetic phenomenon had to have been from an evolutionary point of view, it's very highly conserved, and you try to begin to think of it within an evolutionary framework for survival advantage, and it really has not received the attention it needs from, say, really hardcore evolutionary biologists, because this is, I think, at the core of life going forward, that sort of thing. So hormetic responses, in my opinion, they're integrated responses across multiple systems of biological organization. So say, for example, you're looking at cell proliferation, or it could be anything. You might have a cell that's enhancing cell proliferation, but when you look at people that have done detailed studies on some of this, you might find, gee, this chemical that's inducing a hormetic response, well, it may be activating 700 genes and deactivating 500 others, and these get all integrated in some very complex way. And the end result of this interaction is a biphasic dose-response relationship. There's a lot that's going on in all these operations, but somehow there is this integration that results in the limitation of the response to this 30 to 60% range that affects what is necessary to enhance survival and the proper allocation of biological resources. But this is happening for all of these endpoints. If you think, well, this is just for cell proliferation, well, it's for fecundity, it's for tissue repair, it's for learning behavior, it's resistance to disease, it's tumor incidence, all the threats that we have for aging and longevity. You see the same pattern happening, and each one of them has their own level of massive complexity, but yet it all gets reduced to a simple dose-response relationship that becomes very predictable with a lot of applications. And yeah, And so this is just, I'm going to show a few, a handful of examples that are, some are in vivo, some are in vitro and so forth. This is one this investigators from Binghamton University, New York, did this work. I invited them to a conference, and there was a behavioral study showing the effects of ethanol on social behavior in their adolescent rats at postnatal day 30. And it was so interesting, you know, at high doses, these were almost like comatose, quiet, and so forth. And as you drew back on the dose, and you followed, she presented her findings on a videotape, and you see a beautiful inverted U-shaped dose response. I went out to her place and visited their research, but it followed the ethanol behavior is following a nice hormetic dose response. You see the 60% increase at maximum. Here we have a different endpoint. It's a longevity endpoint, this time with fruit flies with a very toxic agent called methanol. And at high doses, it's shortening the lifespan of these fruit flies substantially in a linear dose response. But when you bring it back up towards the control value, the dash value, it flips to the other side, and you have this stimulation of this enhancement of longevity. And this has been reproduced in a number of different labs across the world. It's a reliable response. Here we're looking at alixin. This is alixin's a component in garlic, and here we're looking at the primary cultured hippocampal neurons in the E18 of the Wistar rats. And you can see this biphasic dose response conforming to the hormetic perspective. Here we're looking at sodium arsenate on lymphocyte stimulation, bovine lymphocytes. What I like about this, but the others are there too, is that you see all the doses that they use. I mean, about 12 doses or so. I don't know how many there are here, but you can see that this kind of goes back up as the dose gets lower. You're approaching the control group, and then you have the flip to the other side, and you have this typical hormetic sort of a dose-response relationship. Most people, they don't study this many doses, and they don't really go low. I mean, you see something at high doses, and you'd normally just extrapolate back to the control value. But actually, there's biological activity going on below that. Here we have a really toxic agent, methylmercury. This is like one on EPA's, top list. Methylmercury is a neurotoxin. Pregnant women are advised to avoid fish with methylmercury, things of that nature, for obvious reasons. But if you take this neuronal cell line and you expose it to a wide concentration range of methylmercury, at high doses, it's pretty sharply harmful. But at low doses, it actually stimulates the viability of these nerve cells. It's in a hormetic fashion. Here we have nicotine on human osteoblast cells, this MG63 cell line, and it's showing this biphasic dose response. I have these white blobs on this, and the white blobs are put there because these people tried to use, and they were effective in using a certain receptor inhibitor at this concentration to see if they could block the stimulation, to see if the stimulation was mediated by that receptor. And in this particular case, they were actually able to block the stimulation by a particular receptor inhibitor at these two locations. They were unsuccessful in reversing the inhibition response. So it was saying that the stimulation was being mediated by a particular receptor, but that receptor had nothing to do with its inhibition. Now, different investigators do different things, but the point here was that the nicotine was having a biphasic response on this cell-based endpoint. Here's looking at inducing seizures with this agent called PTZ, and in general, what people are really trying to look at is can a drug make the toxicity that's induced by the PTZ reverse it a bit? Because they're trying to look for therapeutic agents. So you can see a bump that goes up in the middle, so to speak. It's telling you that this agent has therapeutic potential. So in this case, they looked at morphine, and they found, wow, there was this biphasic dose response at a lot of concentrations they looked at. And you can see around 30% increase. And so it was saying that this compound was able to reverse or prevent some of the effects of the PTZ induced on seizures. And it suggested that morphine had some potential, but it was a biphasic dose response. this got me interested, and I ended up finding about 75 agents in the preclinical literature, all different agents, but within these PTZ and related seizure models, all showing this same sort of a dose response relationship, so that most anti-seizure drugs preclinically were showing a biphasic dose response. They were actually almost all acting hormetically. I published a paper on that a number of years ago. And this is the same sort of an idea, but now it's looking at anti-anxiety drugs, where you have the mouse, and the mouse doesn't want to go and wants to stay in a darkened zone, but if you can try to get it to go into do something it doesn't want to do, like go in an open area, it's something that obviously wants to avoid, doesn't want to get picked up by a hawk or something like this. But if you can get it to it's a sign of anxiety. But if you can get it to go into the lighted areas, open areas, it's a sign that you can reverse or prevent the anxiety. And in this case, the time spent in the open area, you can see goes up and acts hormetically, and then goes down at higher doses. They become even more anxiety filled at the higher doses. And I went into this literature quite a bit as well, and found really over 200 or so different drugs in the preclinical literature tested for anxiety that all showed these biphasic dose responses. And then in the animal studies, preclinical, then they'll choose a dose or two in the middle and test those in the clinical trials. Some make it through, and they may be used to make money for the pharmaceutical company, but they're all based upon a hormetic concept, even if they don't know that, but it's true, in my opinion. This is an example of resveratrol, which has had a lot of attention, and looking at its effects on myocardial infarction in an animal model and cardiomyocyte apoptosis. And you can see the J-shaped dose response at low doses. At high doses, it actually makes things worse. Here we're looking at a whole bunch of I call plant-derived agents, looking at their inflammatory capacity. And these are plotted, their extracts are plotted. And you can see they all act so they all have their own unique dose-response relationship, but they all follow this biphasic dose-response pattern. Now, one of the important areas of hormesis is the idea of preconditioning adaptive responses. And numerous agents have, if you give them prior to a stress or after the stress, then they can display, and they often do display, hormetic biphasic dose response relationships with mechanistic explanations. This is true for low doses of ionizing radiation. It's true for almost every kind of chemical in cell. And so there's an awful lot of work that's being done with respect to preconditioning and hormesis, less so with post-conditioning, but they also respond in the same fashion. I'm going to jump over here to show you some cancer endpoints. This is a study that was done at Oak Ridge National Labs and with gamma rays on mouse lung adenomas. And each one of these little triangles or squares has about 500 to 750 mice in them. It's a gigantic study, just a gigantic study. And you can see that as the at high doses, you have the beginnings of the increase in tumors, but at low doses, you have about a 20 to 25%, 30% decrease in these adenomas consistently for males and females following a predicted hormetic pattern. Over here, this is a study from Japan looking at these DB mice, which are predisposed to developing diabetes. And this is showing you that at 90 weeks, there were 70%, and they're being exposed to continuous radiation, which is about 70 to 80 times higher than background radiation. And so it's high, but not too high, but it's consistent every hour. And after 90 weeks or so, the treated group, 70% were still alive, and the control group only had 40%. But what I want to show you is, it comes up next, and that's a representative of both of the control group and the treated group. The control group, it looks like my dad did, who died at 95, just before he died. He was kind of really well worn, lost his fur, kind of frail, and just did not look healthy. And that's kind of how these control groups look. This is a representative of the control group. This is a representative of the irradiated group. it looks kind of pleasingly plump, shiny fur. It looks like it's got a lot more life in it. And it ultimately lived about 30% longer than the control group. You'd think that was going to live forever. It didn't live forever. It lived about 30% longer, but it had what they call a healthier lifespan, in theory, and it also lived longer. If you didn't know it was radiation in that, we had a sign up there saying, well, gee, this was the resveratrol group. You could run out and buy resveratrol. But this is an example of and I saw this in two ways. You might look at it differently, but people would look at this from a hormetic point of view, that low doses of radiation seem to be beneficial. I looked at this as living the good life wasn't so good. And what was the problem with living the good life? Because this is the life that I probably live. I live the life that EPA tells me to live, more or less. I don't smoke. I don't do this to excess. I don't do this. I'm living the control life, and I'm saying, okay, we know a lot about normal metabolism, and normal metabolism will produce the amount of oxyradicals produced by background radiation is outdone by about 2 billion to 1 for background metabolism compared to what background radiation does. It's our worst enemy in many ways is the person we look at in the mirror. So this is interesting. So the effects of gamma rays on lifespan in the female house cricket, it follows the same pattern. High doses are harmful, low doses you get the flip to the other side. It's still in this 150, 160 zone. Over here, we're looking at the frequency of sex-linked recessives as a function, in terms of mutations, as a function of gamma ray dose to immature male germ cells of Drosophila. It's the same model that Hermann Muller used, that he got his Nobel Prize in 1946 for showing that X-rays caused mutations. But he used very high doses. If Hermann had decided to do a broad dose response, he might have then detected the J-shaped response, and he could have got 2 Nobel Prizes, one for inducing mutation and the other for showing the anti-mutagenic effect at low doses. Okay, this is DDT on liver foci on male Fisher 344 rats. DDT has been banned as a carcinogen, has other toxicological issues, but it causes liver cancer. Japanese investigators decided to do a big study looking at a marker for liver cancer, these liver foci, with a lot of doses. And what they found at high doses, there was a high correlation between the foci induced and the occurrence of liver cancers. But at low doses, there was this distinctly hormetic dose response. It's a J-shaped response that these investigators did a meticulous job at trying to figure out the underlying mechanisms for protection at this level. And what they're really showing was the generalizability of this to just, this agent, this endpoint, and this model. Over here, we're looking at something called the Mega Mouse study. This was an FDA-funded study, had 24,000 mice, and they were really trying to figure out the nature of the dose response in the low-dose zone. And they used this well-known carcinogen, acetylaminofluorine, that causes bladder cancer in this model. And what they found was that there were so many mice that they had to use 6 different buildings, all different setups. And what they found was in each building, they had all the dose responses represented. They just had to sum all the data. They found in every single building that the dose response was J-shaped. And they found that, yeah, it would cause bladder cancer at high doses, but at low doses, it actually decreased the risk by about 40%. And this is called the effective dose 01 Mega Mouse study. It's a very major toxicological study. So what is hormesis actually indicating? In my opinion, this low dose stimulation is different than high dose inhibition or toxicity. The low dose stimulation, in my opinion, it's a measure of biological performance, not toxicity. It determines how much a system can respond. Even when you're looking at chemical mixtures, and toxicological mixtures, there's almost no endpoint, no limitation to how much can become toxic, or how much could it be enhanced. But when you have mixtures of hormetic acting agents, even when you pull them together, they still do not exceed this 30 to 60%. It's the concept of synergy. Hormetic synergy is really different than toxicological synergy. And again, I think it's constrained by the limits of what I'm calling biological plasticity. So the low dose, as I said, the low dose stimulation to measure biological performance, it really determines how much a system can respond. Hormesis is the first quantitative estimate of biological plasticity. The hormetic stimulatory response is constrained by the limits of plasticity. I haven't seen anybody break through the limits of plasticity, and I've looked at a lot of examples of this and trying to figure out how to do it. Hormesis has a lot of application, especially in the world of big pharma to anxiolytic drugs to anti-seizure drugs, memory enhancement, osteoporosis, wound healing, and multiple inflammatory conditions. About 3 or 4 years ago, I decided to spend some time just on stem cells and hormesis and ended up doing about 14 papers on it. With every stem cell that is out there has copious amounts of hormetic dose response relationships within them. And I have to say, most of the people who report dose response relationships that are hormetic, they have no idea that they're working with hormesis. It's very hard to find hormesis examples going into PubMed, Web of Science, using the terms hormesis or hormetic. You'll be very disappointed with what you find because the people who are reporting these biphasic dose responses, they don't know even how to characterize them with a term. It's really very interesting, very challenging to an investigator. Now, it has medical applications. It could be very useful in how you could apply it to organ transplants, to enhance surgical success, to enhance the patient care and chemotherapy and radiation therapies. People who deal with intermittent fasting and so forth, they've related these dose response wise and otherwise to hormesis, same with exercise and so forth. It's built into every system and activity that we have. So in general, some conclusions for this is that hormesis is a general and central biological concept, in my opinion. It affects all disciplines using the dose response, and that means it probably affects all disciplines. Now, it represents a general adaptive strategy through which biological performance is enhanced and mediated. Hormesis provides both the theoretical basis and the molecular foundations for creating biological shields. And that's a really significant concept. And these biological shields can protect us against chemical, radiological, and microbiological threats. And we can throw in the biggest threat that we have is aging in many ways. And how can we use it to slow down the process of aging and all the other decrements that go along with it. This has implications for numerous and diverse medical interventions, as well as affecting health and performance of military personnel, athletes, the general public, including patients, accident victims, and others. And one of my conferences I had a person who was dealing with shock, and he was removing blood from these animals and sending them into shock and trying to see if he could protect these animals from shock by giving them, hormetic stimulation, and he was very successful. And then was trying to apply this to the real world, taking motorcycle accident victims to the hospital. If you could intervene with hormetic interventions on the ambulance going in, how could you enhance survival and things of this nature? I don't think there's an endpoint that you can't enhance through a hormetic application. Failure to consider the hormetic dose responses can lead to improper dosing, resulting in possible ineffective or potentially harmful effects at low or high doses, respectively. From my perspective, which is historically been strongly environmental, failure to consider hormetic responses within hazard assessment and risk assessment has really been a serious failing of modern risk assessment. Hormesis can and should play a significant role in drug discovery, development, and evaluation. And hormesis should become fully integrated into educational programs for all biomedical and medical students, continuing medical education programs. I'll end with just this little crazy little story. 3 years ago, I was giving this talk to my students at UMass. This was like a master's program group. And there probably 40 people in the classroom, and they had been there most of them had been at UMass and stuck around for a 1 or 2 year post-undergraduate situation, getting a master's degree. And so I asked him in class, I said, how many of you have heard of the term hormesis? And none of them had heard the term of hormesis. I said, you know, you have, I said, this has been my life. I said, and nobody in this class has heard of the term hormesis. I said, well, you know, I said, just to let you know, there was a nice big paper in the journal Cell a few years ago that listed the eight hallmarks of health, and one of them was hormesis. And you guys are in the School of Public Health, and you never even aren't even aware of that. I said this educational process is a total failure. And so, of course, I was called down to the dean's office after saying that the school was
Oh,
terrible or
yes.
whatever. But in any case, that's the end of my story. I'd be happy to take any sorts of questions that you might have. I will.
Ed, that was such a fascinating talk, and we'll have to have you come back for questions. I'm sure there are a million questions that people have based on all the information you've given us. But I guess we are past time. Maybe
sorry.
one question. Does anybody have a question they want to ask Ed right now?
First of all, that was great, Ed. It's such a fascinating topic, and it's so great to hear it from the guy who really helped create the whole field. And we use a lot of brain organoids and 3D culture models to try to do drug discovery for Alzheimer's and in our unit. And I'm wondering if there's some way, and I know it must be an answer and conversation we don't have time now, but I'm wondering whether there's some way in these organoid models we could test something that is toxic at high levels, but might make the neurons more healthier, and reduce pathology at low doses. Just might be worth thinking about modeling this in the organoid models, because it takes too long and a lot of time in mice. But just a thought.
Right. Now, I think this is hormesis, and in this direction would be a really good interface, and I'd love to talk with you about it.
Yeah.
Yeah.
Great. That's wonderful. Ed, thank you so much for doing this for us.
And I'm sorry I'm so wordy.
No, it's really fascinating. I saw that pretty much everyone stayed for the
that's
entire
cool.
talk, so it speaks volumes. And we'll talk again. We'll
Thank you
keep the
guys.
conversation going.
Thanks for the invitation. It's a beautiful day outside, so make sure you get a few minutes out there in the sun.
A
Okay.
few ionizing radiation.
Yeah. That's good. Make sure you get a little bit of sunlight today.
All right, very good.
guys hang in there.
Gene Bowman mentioned trace metals in the comments, and that's interesting because of the Ayurvedic use of metals at low doses. That'd be an interesting thing to look at.
Oh, that's right. Yeah.
Yeah, lots of follow-up questions. Would love to have you back, Ed, for part 2
Well,
Yeah.
at a seminar series for discussion. Lots of comments.
Yeah.
Talk.
well, thank you very much. Have a wonderful day.
Wonderful presentation.
Nice to meet you all too.
Thank you.
Okay.
Okay, bye-bye.
Bye.